Morning, everyone. For those of you who don't know me, I'm James Moore. I'm the head of the Department of Biology. It's a real pleasure to welcome you all here today, friends and co-workers and family of Simon. It's obviously very bittersweet that we're here because of the tragic death of Simon. But I think it's a real testament to Simon's brilliance as a scientist and as a human that so many people have given up their Saturday morning to come and be here and I hope to celebrate the great achievements that Simon made and have a bit of fun on the way. I think hearing some good stories. I've already seen some bad language in the slides that are coming up, so looking forward to that. And to reflect a little on a life well lived, if cut short. So it's really great to welcome you all here, and thank you for those particularly who've travelled a decent distance to get here as well. I would like to thank Erin and Claire, who've probably disappeared somewhere, but they've been organising the event today, and they've done a terrific job with that. So thanks to Erin and Claire for what you've been doing. Erin sat up the back. I can't see where Claire is.
Up front, outside.
Yeah. Yes. On your bits of information about the day, there's a schedule for what we're doing, and on the reverse of the sheet is something I'd like to say a few words about, which is so those of you from biology will be very familiar with some of the schemes we have to support students to get research experience within the department, and we have this scheme called Generation Research, which encourages people from non-standard backgrounds who are perhaps underrepresented in research to get an opportunity to experience research and to help kind of widen the breadth of backgrounds of people in the department doing research. And one of the ways we do that is through a summer studentship scheme, where postdocs can write projects, and we recruit students, preferentially those who come from a less advantaged background, into summer studentships, which often lead to really good experiences for those students and a future in research for many of them. And we'd like to name that scheme after Simon going forward. So it'll be the Simon McQueen-Mason studentship scheme, which I think is really fitting given the unusual path to science that Simon himself took, and his and the joy that he took from that, and the joy that he brought to so many people who he trained through his research work too. So there's the scheme, and this is funded through departmental funds, but we're always keen to try and build up more opportunity for more students. And so there's a link here to a giving site, and if you're interested in that, please have a look, and if you'd like to find out a bit more about it, I'll be happy to talk to you at one of the breaks today. I've got a thing about practicalities where I have to tell you that there's not a fire alarm. Let me see if I can find it. And I've got to. This reminds me I need to switch my own mobile phone to silent in a minute as well. So if there's no scheduled fire alarm this morning, so if the sounder goes off, then you'll need to leave through the fire exit. Please be aware that the AV team will be video recording, so remember that when you're swearing loudly. There'll be refreshments and things outside after the break, but I realise that we're already running massively over time, and so I shall hand over now to Neil.
Thanks, James. And yeah, I'd like to reiterate what James says. Thank you all for coming. It's lovely. We've got so many people who've turned out on a Saturday morning. And if you're here for some hardcore cell wall science, I'm afraid you're going to be disappointed. But you will hear some of Simon's inspirational work that he's done. And yes, you'll hear a few anecdotes along the way as well. So our first speaker, in fact all our speakers have worked closely with Simon. They're good friends and colleagues and collaborators. And many of us have obviously worked with Simon for many years. And the first of our speakers is Daniel Cosgrove, who was Simon's PhD supervisor. And Simon spoke very warmly and often spoke about his time at Penn State University and doing his PhD there in Daniel's lab. And I had the pleasure of meeting Daniel when he came over to see Simon and visited CNAP. And I've had some correspondence with him about this meeting, and he was telling me that that Simon, he always used to say how inspirational Daniel was as a scientist. And Daniel said to me, actually, towards the end of my career, it was the other way around, and I looked at Simon as the inspirational scientist, which I thought was lovely. So obviously Daniel's not here, he's at Penn State University, but he's kindly videoed a presentation for us. So, thank you.
My name is Daniel Cosgrove, and I served as Simon's PhD mentor at Penn State University more than 3 decades ago. Here I want to recount some early aspects of Simon's work on plant cell walls. When I first met Simon, he was already working in a neighboring lab on the biosynthesis of a plant hormone. But before he could settle into that lab, the graduate program required that he complete a rotation in another lab. So together we worked out an exploratory project in which he would try his hand at extracting proteins from plant cell walls and testing them in what we called a creep assay. This was very exploratory, and I anticipated he would try it for a month or two and then move on and move back to his original lab. The work was in the context of understanding molecular mechanisms of plant cell growth, which was a topic that Simon was interested in. It turns out the initial results were so promising that Simon joined my lab to work on this project for his PhD dissertation. This resulted in a series of groundbreaking publications, the first of which is shown in my first slide. Please bear with me while I switch things over. All right, so that paper is shown here, published in the Plant Cell, announcing the discovery of these proteins that induce plant cell wall enlargement. It's been cited more than 1,000 times in the scientific literature, and even after 30 years, it's still garnering about 40 citations a year. Simon's work has impacted our understanding of several aspects of plant biology, but before I talk about these, let me show you a couple photos of Simon in these years. Here's Simon in 1992 in Pittsburgh at a conference where we unveiled expansins for about the first time. This look on Simon's face is very characteristic of him in those years at least. And I thank Florencia for sending me this and a couple of other photos of Simon. Here's Simon with Florencia and baby Ian. This is back in State College a little bit later. I looked hard for photos of Simon in the lab. This is the only one that I found. I call this Simon Through a Glass Darkly. You can see Simon's huge grin. He was fun-loving and he enjoyed pranks, and I was a target of one particularly effective prank, which I want to recount. In the early days, when we were still isolating expansins, I was in Germany for a time, and we communicated by phone and by snail mail. In one of my weekly mail packets, I found a poor quality photocopy of what appeared to be an advertisement from Science announcing the sale of wall loosening peptides. This turned out to be Simon's hoax. Seeing this advertisement got me excited and fearful that we were scooped, and so I immediately called the lab to query Simon and my technician, Dan DeRoshko, who was his partner in this crime. They had a good laugh and admitted their antics. And maybe Simon was thinking of this hoax when I took this photograph. See that big grin on his face? That seems so characteristic of Simon. So a series of papers followed from the first, where Simon characterized the activity of the proteins, ultimately leading to the cloning of the expansin genes. And this paper, I was very proud when Simon got a research position at University of York and was recognized with the President's Medal by the Society of Experimental Botany with an invitation to summarize his work in this review. And this review is a nice summary of basically his PhD work in my lab. Simon subsequently made several other important contributions to the cell wall field, but I will limit my comments to his work on expansins, which had a foundational impact on our understanding of plant cell growth and various aspects of cell wall structure, and also some aspects of biotechnology of biofuels. I want to tell you how all this started. We knew that growing plant cell walls possessed a property called acid growth. That's shown here. If you prepare a cell wall specimen from a growing stem, clamp it under a device called an extensometer, clamp it under tension, what you find is that the wall is not extensible at pH 7, neutral pH, but very quickly starts to extend when the buffer is changed to an acidic buffer. So this is acid growth. Heat treatment eliminated this pH-dependent activity, and so we hypothesized that there were wall loosening proteins bound to the cell wall. Question is, could we identify those? Simon's breakthrough was to develop a robust protocol to extract the proteins and then add them back to this assay system to measure creep at the cell wall, as cartooned here in this diagram. This worked and led the way to all the subsequent progress. The tough work followed, in which Simon then had to separate the expansin protein from all the other proteins in the extract and identify its activity using this creep assay. So this was a challenging on many technical levels, but Simon's persistence and hard work and good judgment, as well as good luck, resulted in the success of this first stage of the expansin field. That then enabled many follow-on studies in many labs around the world. Some of the topics are shown here in this figure. Simon's work is at the base of this branch. Subsequent work on expansins showed them to be involved in fruit softening and in the activity of microscopic worms, pathogenic worms called nematodes that attack plant roots, as well as in several other phenomena shown here. The cloning of the first expansin genes by Simon and collaborators led to studies of expansin gene regulation under the control of essentially all the plant hormones, various developmental stages, and a number of environmental conditions such as light and water deficits. It also enabled a better understanding of expansin genes, the genomes, and gene evolution. And Simon made a key contribution to that aspect of the work with this paper shown here. This ultimately led to the discovery of expansins in bacteria, and they are much more prevalent than what we thought before. Turns out many of the plant pathogens have an expansin gene, and we think that they use these to help their invasion of plant tissues. There's another class of bacteria that are thermophilic bacteria that break down cellulose using an enzyme complex called a cellulosome. This is a device that they attach to the surface of their cells, these bacteria, and it then interacts with cellulose and very effectively breaks it down and chews it up. And this has been used in the biofuel field. There's also active work going on around the world to engineer improved cellulosomes using expansins. Lastly, expansin has been used to modify plant traits. One example comes from Simon's group in 2020. This was an attempt to increase wheat yields by staged selective expression of expansins, and it proved successful. I thank Ian for this image of Simon. His pioneering work on expansins opened new vistas in plant biology and biotechnology, inspiring many to dream of new possibilities.
Actually, there's got some quite detailed science in that one, but I think that's probably about as much science as you're going to get today. But yeah, I'd like to thank Daniel for taking the time to produce that fantastic video. We were just joking in the car coming here that Simon's a brilliant scientist, creative, and he just sort of ploughed on doing things that he wanted to do, and at times just didn't realise the chaos that he created around him. And he had this sort of brilliant team. There's Veronica, Claire Still-King, and Alexandra and others, but in particular Leo, who's giving the next talk. And so I'll hand over to Leo Gomez. Oh, there you are, Leo. Yeah.
Okay, morning everyone. I'm Leo Gomez, and I've been working with Simon since... Thank you. Just in case, there was someone in the room that didn't know that. And I've been working with Simon since 2006, and I met Simon in 2003 when we moved to York. Actually, I met him in 2001 in Buenos Aires, but he couldn't remember that, so, let's ignore that. But I remember that in 1993, I was doing my PhD in cell wall elongation, and I read that paper that Dan just mentioned, and it just blew my mind that this guy with a long surname had that idea, and I was deeply envious, and, little I knew that I would spend the vast part of 20 years working with him. So yeah, it's been good fun. So I think the no swearing just cancelled half of my talk, so it's going to be faster. So I had to write the bio of Simon, and I didn't feel comfortable. So I went back and looked into his presentations from a few years back, and this is the slide that he used for a presentation for the Royal Society about his life. So he said, he left school at 17 with a handful of O-levels, and then traveled to the US, worked in boatyards in California, and then to Australia and Southeast Asia, worked as a fisherman, bought his own boat, which is that picture that I took, is a still from a video of Simon in the Royal Society website. And then in 1984, at 26, he was accepted to study biological sciences at Portsmouth and obtained a first-class degree. Studied for a PhD with Dan Cosgrove in Penn State. 1994, he was awarded a Royal Society University Research Fellowship to work at York, and that was extended to 2002. 2001, he became chair in material biology at the CNAP. 2014, he became the director of the CNAP, and the cheeky line at the end, he still owes his A-levels. That's a cheeky detail that he would put always there. So that's his bio. Simon in his own words, but in every conversation that we have about Simon, and last night we had a few things come up all the time, and for me these are the most important ones. So his scientific creativity. He worked, he was a plant scientist, but he worked in insects, he worked in food, he worked in industrial biotechnology, in textiles, in seaweed, with engineering of processes, and I think the last crazy project is still running, which is the railway project, where we're looking at the leaves on the tracks, and we're still running that. But it's, it was like immense, the diversity of the work he did. And his vision, about applying that knowledge on cell walls to tackle climate change. And, he would implement technologies or try to implement technologies that would deal with big problems. He wouldn't take the small problems. He would, solve all the problems in the world at once. His generosity, he would acknowledge, I can see people in his team nodding, because he would acknowledge the work of everyone, and he would support the careers of the people around him, and he would give letters of support to absolutely everyone around and go the extra mile. His compassion, he would never walk away from problems. That could mean a beggar in Delhi or someone in the group with personal problems. He would always be there. So that was absolutely amazing. And my favorite, his sense of humor. We had a lot of laughs during the years, and I think that sense of humor is what brought people around him and allowed to run large projects where he would bring a lot of people and put a task in front of us, and we would go for it, no matter how hard that was. So he had that capacity to bring people together, and that could be seen in the many projects he led. And these projects dealt with mostly converting agricultural waste into things that had value, from energy, biofuels, feed, and fertilizers. And these projects reached a number of countries and a number of groups around. So I chose the first one, renewal, which I think it was the transition from perhaps the academic cell wall studies into the biofuels area. And this project had the aim of converting lignocellulosic biomass, that for us was cell walls with a different name, into sugars, and then from sugars into fuels. And I think there's a few people in the room that were part of, yes, Paul, part of renewal. And last week we had the BBNet meeting, and Wout Burden from Belgium was there. He could trace a straight line between the ideas generated in renewal and the 15 years that he had discovering the lignin pathways and the effects on the digestibility of plants. And so this is a bit of a historic document. So this is the first scheme of the renewal project that we draw on his table. On the back of a printed paper, you can see work package 1, gene discovery for saccharification potential. Work package 2, 3, and 4 were obvious: cellulose, hemicellulose, and lignin. And then we would engineer the genes that we discovered into plants and get plants that are easy to process in an industrial context. And that derived in a really large and complex project that, I still call it like the dream team of the cell wall in Europe, because it involved the best groups in Europe working in cell wall at the moment. And that's the team. Poppy and Cara were part of the team when we developed the platform for analysis of cell walls there. And yeah, these are good memories of a lot of meetings. You can see Simon Turner, Claire Halping over there, and Paul Dupré. And it just really generated an incredible amount of scientific ideas. So that was one crucial project. The second one I chose came after that, and it was a really large and complex project, because it was a European consortium coordinated with a Brazilian consortium equally large. So it was twice as large and complex as renewal. And this was, again, in trying to produce biofuels from sugarcane, maize, and miscanthus, and there were a really large number of institutions inside this project. And this project was really interesting from the point of view that we would have 2 meetings every year. One in Europe, that you can see was very formal, You know, we would go to museums, that's Simon holding the EU flag. You know, it was only science. But also, we had every year a meeting in Brazil. So that's a place, Maresias, where we had these meetings. So the meeting was there, and the coffee break was there. So it was really easy to go from one side to the other. That was absolutely awesome, because that you know, environment brought the creativity and many ideas and many points of cooperations between the two teams, and I think a lot of papers and excellent projects came out of this very large consortium, Sunlead. And you know, we had formal meetings and other institutions involved, but also there was a lot of fun, and I had to go through hundreds of pictures to get one that I could show today. But you know, it was really good fun. And also that meeting was by the sea, and Simon loved the sea, so that's Simon. At about 6 o'clock in the morning, it was a storm. He would pretend he didn't understand Portuguese because the lifeguards wanted to prevent him from going swimming, but he would go swimming anyway. So he really loved that place. And another project that ran for a number of years is a cooperation between Vietnam, the Philippines, and Thailand also. I didn't put that there, which was on rice straw. So rice straw is a serious problem because the burning of rice straw produces contamination and also health problems. So if we can do something with the rice straw, that is fantastic. So we worked for a number of years, and that is still running. There was a project that finished in December, and we are still collaborating in a new project with Vietnam on valorizing wheat straw. And you know, one of the main outcomes was to see that the varieties that we selected with high digestibility were being developed in Vietnam and in field trials to get rice straw that can be composted or given to animals as feed. So, and that took, you know, the cooperation from, so that's in the north of Vietnam, and there's people from the Philippines and Thailand there also, to other parts of Vietnam, such as Ho Chi Minh City, Can Tho, and again, a large number of points of contacts and ideas being developed there, and that allowed, you know, the classic lab visits, and also to enjoy one of Simon's favorite cities, that was Hanoi, and that's his favorite bar, where, you know, when we went a number of times to enjoy a few beers and have long conversations about life and beyond there. So, and finally, a project more recent that ended last year, which is a cooperation with India, and the idea is to reduce industrial waste from sugarcane. And the crucial thing about this project was the large involvement of industry. So the idea was to take these ideas from the lab into the large scale and develop industries around the production of chemicals derived from biomass. So that you know, meant a series of visits to sugar cane plants, and also that cooperation is still running. We were in India with Neil a couple of weeks ago, and that continues. So all these projects come to the present and have produced really crucial inputs in academia and also in industry. So yeah, that's all from me. I think we've been, you know, really lucky, and we had the privilege to work with Simon, and it's been absolutely fun. And you know, I will always be grateful for being part of this. So yeah, thank you.
So Leo mentioning Simon's sense of humour, and Daniel Cosgrove as well mentioning as well. I thought I'll just mention, you know, something that, a prank that Simon used to do quite often. I'll be walking with him along the corridor in the department. So this is the director of CNAP, senior professor, in the department, and we'd be walking along, and he'd knock on someone's door, then run off. And then, whoever it was would come out and say, "Yes, Neil, what do you want?" And he's gone. Yeah, so, 50-odd-year-old man still doing knock and run on people's doors. So our next speaker is Claire Halpin, and Claire's sort of background is in plant cell walls. She's a professor at Dundee, and worked very closely with Simon over a number of years, and shared a number of adventures with Simon and myself and Leo. So where are you, Claire? I can't see, there you are.
So I tried to prepare this talk this morning and then found that I just went through all the displacement activity possible. So it'll be rather off the cuff, and I hope I don't ramble. Tell me if I'm going on too long, Neil. So I thought I'd start with, this is a picture from that video that Dan, I think, mentioned, and Leo also mentioned, that Simon had won the Royal Society University Research Fellowship in about, when would it have been? 2000 and, I'll work it out in a second. This is when I first heard of him, though. I was working at Syngenta, or Zeneca Seeds as it was at the time, and somebody actually shared Simon's URF application with me in complete confidence, and I had read it because I was another cell wall biologist. And I was completely blown away by some of the work in it, because not only was he using plants to actually isolate the expansins that Dan talked to you about, but he also had some really neat experiments where he just actually used sheets of paper to work out what some of the properties of the enzyme were. And I was struggling with the problem at that time. We were working with some really woody material and we were trying to grind it down. I was working in a, very well-funded industrial lab, and people were looking for high-tech ways of grinding this material that just wouldn't get down into a powder no matter what you did. And my solution to the problem was to take a joule of liquid nitrogen and hammer into the cold room and just bash it into submission, and that worked really well. So the fact that Simon was using really simple but creative experiments to learn some very sophisticated scientific answers really, really appealed to me. But actually, it was several years after I learned about him and the fact that he was working on cell walls in York when I actually met him. It must have been 6 or 7 years later before I actually met Simon. And that was when we both ended up, and we literally bumped into each other at the champagne reception of a conference in Helsinki. And all of you know how friendly and warm a person Simon was, so it's no surprise that we found it quite easy to chat. It was probably helped by the fact that I knew hardly anyone at the conference and he knew absolutely nobody. And it was a champagne reception, as I said. So when I said that I was going to walk back to the hotel, which was quite a long way, because I wanted to see a bit of the city, he said he'd come with me. And along the way, we passed the harbour and we just segued to have a little look. And there were these two, it doesn't look like it in the photograph, but absolutely incredibly impressive icebreakers there in the harbour in Helsinki. So of course, that was perfect opportunity for Simon to tell me all about his maritime history, and in that very first long conversation I think I learned quite a lot about him. His love of travel, his sense of adventure, his fascination with the natural world, the fact that this was somebody who would take an unconventional path and just pursue things for the hell of it, and even despite those itchy feet, his deep commitment to his roots and to his family. So it was several years really before we actually came to do any work together. In the intervening years, I know I saw him a bit, but I can't remember when or how. I think it might have been at plant biotechnology meetings that Diana Bowles had organised. But actually, we came together again at an EPO Bio meeting in Wageningen, where there was a workshop that was discussing how we could actually, this was really the start of some of this work of trying to understand how we might use plant biomass to make products that would be useful and to some extent replace the use of petroleum. And at that workshop, one of the things we discussed was what was the barriers to this whole field moving forward. And one of the barriers was the fact that we just didn't have a really simple and easy assay for working out how digestible plant materials were. And if we wanted to look across lots and lots of plants and begin to learn something about the genetics of digestibility, we really needed an assay like that. So we talked about it, and everybody went home, and most people thought, oh, it would be nice to have something like that, but it's probably pie in the sky. But Simon and Leo came home to York and they built it. And this is the robot that was in some of Leo's slides. But in retrospect, and I was thinking about this, and I went back and thought about how this whole field has evolved, I think that was really quite a seminal moment. It was unique in Europe, the platform that they built, and it really underpinned a lot of the work that I'm going to talk to you about afterwards. I have very few data slides. This is just one of them, and this is just to kind of reflect, to some extent, Simon's generosity. I mean, he and Leo had built this platform, but he invited me in to give him some materials, some plants that we had genetically modified. We'd modified genes in the lignin pathway in these plants, and we would expect them to be a lot more digestible because of that, because lignin is one of the barriers to digestibility. And they were just able to show that by putting these on the platform, you can see in the histogram on the bottom that you could indeed see that compared to normal plants, the wild type WT, these transgenic plants, you could release sugar from the cell walls much more easily. You could get twice as much sugar out of some of these transgenics, which just reflected the fact that they were more digestible. It was easier to get the sugars out, and the sugars are the components that we can then use to make other useful chemicals from. So this platform became very important then in other big projects that Simon and I were both involved in. Leo's already mentioned Renewal, so I'm not going to say too much about that. But another one was the BBSRC Sustainable Bioenergy Centre. And just in case anyone doesn't really get how and why we might want to use plants to make fuels or energy from, I mean, at the moment, you know that we get our fuel and energy by mining oil and coal and other fossil fuels that, when you think about it, actually are the dead remains of plants that have been buried and compressed in the ground millions and millions of years ago. And in mining that material out of the ground, we release a lot of carbon dioxide into the atmosphere. Carbon dioxide that was previously locked away, safely locked away. And of course, when we burn that oil or coal in our cars or in our houses, we release even more CO2. But plants actually take CO2 out of the atmosphere and lock that up in their bodies, in their biomass. So if we were to take plant material and instead of having an oil refinery, have what we call a biorefinery, where we use that plant material to make fuels or to make chemicals, then the whole thing could be carbon neutral. And that's the whole idea behind a lot of the work that Leo talked about and that I'm just going to very briefly mention now. So in BSpec, there were 6 different component projects where people were working on different aspects of this problem, from growing biomass crops for the UK. There were 2 on, myself and Paul Dupre led 2 here on different components of the cell wall and learning more about those. There were 2 over here. Nigel was leading one of them, about how you would, once you got the sugars out of cell walls, how you would use bacteria to make useful products from them. But in the middle here was the one that Simon had with Simon Cragg, also in Portsmouth, where again they had a really novel idea of using marine organisms to try and isolate really novel enzymes that could be useful in this process. And I'm not going to say any more about that because I'm sure you'll hear about it later. All I'm going to say is that the rest of us were always gutted when we were talking about this project, that the only thing that people were interested in was these sweet little organisms called the gribble. But again, it shows his creativity and his inventiveness in coming up with that project. And what I did as part of that project, I just want to pay tribute again to the development of this robot and how it completely set up all the work that we were able to do to try and understand some of the determinants of digestibility. We grew hundreds and hundreds of varieties of barley. Then they were assayed in York for us on the robot. Leo must have done tens of thousands, and Cara and Rachel, who are both here, tens of thousands of samples analysed for us. From that, we were able to work out what the digestibility was of each of those 680 varieties. And then we're able to put that together with some genetic information to tell us where, and you can just imagine these are the chromosomes of barley laid out end to end here. Basically, the data told us where in the genome of barley there might be, you know, in red here, genes that were important for digestibility. And of course, these were in 650 different cultivars, but having this information now allows breeders to take those useful gene variants and try and bring them all together in a single cultivar, which is how we will improve plant biomass for some of the kind of processes that we're talking about. So, you know, those were two big projects. Leo talked about others, and I think it was during the course of those that Simon really began to emerge as a leader, not just within the projects, but actually nationally in this whole research area. He was often the person who was asked to come and speak, both to the public or to politicians or to industrialists or to other researchers, partly because of his great enthusiasm and knowledge in the area, and partly just because of his great personality, I'm sure. And as Leo said, this led to a couple of big UK-wide networks that he led, including BBNet, which is still ongoing and that Neil is now leading. But as well as the UK effort, Simon's perspective was always global, and this is where some of the trips to Vietnam came in. And I remembered as I was putting these slides together, the very first time that we went to Vietnam, we were all put together in a room with people from a whole load of different institutions and politicians and people from government departments in Vietnam. They all sat on one side of the table. They all sat on one side of the table. Now I spoiled my gag. And we all sat on the other, and it was really very peculiar. They all came with different agendas. We had different agendas. We, didn't speak any Vietnamese. They spoke some English, but not always perfectly. And we were trying to work out how we could actually do some work together that would be useful. And I really think that without somebody as relaxed as Simon, but also as, somebody with the tenacity that he had to keep going back and asking questions, that we mightn't have come to a useful conclusion, but we did. Now, I do have a very unique photographic event for you in just a second, and that is Simon in a suit. I don't know if you can really see it because it's quite dark. A suit and tie, and not only a suit and tie, but shoes and socks. And I think this is the only time I ever saw him in a suit. And that just kind of shows you the formality of this meeting. And actually it was more than formal. It was really quite awkward at times because I think towards the end they plonked on Simon the fact that they were going to have a signing ceremony for some document that he couldn't understand and wasn't sure he was really authorised to sign. I mean, in the end, you can see he's looking quite nervous. In the end, I think it turned out there was really just a photo opportunity. But that was Simon. He could carry anything off with aplomb. You just had to sit back and admire it. So I can't remember where I'm going now. Oh yes, and this is just, Leo's already introduced you to this. This is just to remind you why we did it. Of course, it was great fun. It was wonderful to meet people from other cultures. We've made friends for life, but there really was a driving force of trying to do something for good for the planet and for people, because in Vietnam, these kind of cereal wastes are really causing big problems of pollution and health problems. Now, the sartorial elegance obviously went downhill from then. This is actually on the very same trip. I think it was in February, and here he is in sandals and short sleeves, where everyone else is in heavy coats. It doesn't help that Robbie beside him is made in the same mould and just doesn't feel the cold, but yeah. And Leo's shown you many of these. We went to many different parts of Vietnam. We were, I think this was in Ha Dong, and this was in Ho Chi Minh City. And it didn't matter which group of new people you put Simon in front of, he was very comfortable and he was always able to get something useful out of the conversation. That's not to say that he was always prepared, and Neil has already alluded to this. We were talking about it in the car on the way here. There were several meetings where he was completely flying by the seat of his pants, and either Leo or Veronica or Neil or somebody else had to kind of step in and remind him what the agenda was. But again, he always managed, and he did brilliantly. In terms of the sartorial elegance, he wasn't always prepared either. No, you know, one time, for example, they plunked a TV crew on us, or a video crew, in the middle of a meeting, and all Simon had in his bag was shorts and a short sleeve shirt. I mean, he didn't actually even have a pair of trousers with him on that trip, I think. And yet, you know, he carried this off, and I don't know if you can still find this video. It used to be on the Field Crop Research Institute in Vietnam's website. Okay, and as Leah's shown, we went to different places. And I put this one in. I see Rachel's here. You know, this was actually a slightly different meeting where the University of York actually funded, under their GCRF funding, for us to go to Vietnam to try and put together a very large proposal for a big bid for hubs, hub research. And here's Simon starting this meeting with Rachel's support. But also I wanted to use this to illustrate a couple of things. You know, Simon was well able to talk in these meetings and to bring people along with him, but he also really listened. We really listened in these meetings and tried to learn what would be useful to do, what would be useful for the people on the other side. And this also illustrates, although we put a huge amount of effort into this and into applying for money, we didn't actually get the money in the end. And that's something that I really wanted to mark about Simon, was that, you know, he just never stopped. He worked really hard. He never stopped applying for money. He had this ability to assemble really skilled and really valuable and loyal teams of people around him. And I used to really envy that, although now I can see that it was very much because of the kind of person that he was. But he also made a commitment to those people, and he never stopped applying for funding to try and make sure that there was always money there to keep people on. And there were lots and lots of applications that weren't successful. You're only hearing about the ones that were. Of course, they should have been. A lot of them should have been funded. Yeah. And this again was just to remind you why we were doing this. So when we went to Camp Ho on the Mekong Delta, we heard very much about the problems that they have with flooding and salinisation, problems that are a direct consequence of climate change and that they're increasingly struggling with every year. So, you know, these houses here, I think, are normally built on this slightly higher ground between the fields, but these fields should not be flooded in this way, and the water that they're flooded with is now salty water, and so that's causing all kinds of problems. And this is just to show you, I talked about projects that didn't get funded. These were two, though, that did get funded, where we took that same approach of using the robot to look at digestibility and then trying to look at a whole load of different, in this case, rice cultivars. And actually we looked at a whole load of different properties of rice, both in the Philippines and in Vietnam. Flowering time, the amount of biomass they make, the amount of silicon in them, their yield, and we have genetic determinants for all of these now that would be useful for breeders to try and breed better crops. And I'm getting very near the end now. I hope I'm not way over time. I just threw this in to illustrate another thing that used to amaze me about Simon when we'd go on these trips. But I couldn't find a photograph to properly illustrate it. But, you know, we'd be on other people's timetables. By the time a meal was put in front of you, you'd all be hungry. Simon would be starving, you know. And he was always somebody who loved his food. You know, I know half of his family are brilliant cooks. And you'd sit down to some nice kind of starter, and I'd be just getting ready to get into it, and I'd look over. Simon would have inhaled his. It was gone. And, you know, he was sitting there waiting for the next course. I could never understand how he could actually eat his food so quickly. The last project I'm not really going to talk about is the most recent one that I've been involved in. It was called Max Bio. It was one that I led, where what we actually did that was a little bit different here was, instead of us all working on our slightly different areas of trying to improve the plant feedstocks, trying to develop the enzymes and the pre-treatments, and Nigel trying to, before he got eaten by a lion, trying to work on the bugs that would engineer useful products from these feedstocks, we actually quite successfully, I think, for the time of the project, managed to put it all together and go from plant materials to products that were being made in a bacterium. And a lot of this was done during lockdown, where we ended up having a lot of virtual meetings. We all got to know the ceiling beams in Claire's farmhouse quite well, because that was generally where Simon would be. And it's Lee here was actually in lockdown in China for a very long time and not able to come back to the UK. And it's a shame in a way that that was the last project and that we didn't get together to meet in person as often as we might like to. So I've said a few things that I loved about Simon, probably not even the half of it, but like Leo, I think one of the things I enjoyed most about Simon was that he was just a complete goof. You know, you could just have a real laugh. I have no idea why he's got this cocktail on his head. Most likely it wasn't the first cocktail in a coconut that he'd had that evening. But yeah, it's not an exaggeration to say that it was really one of the biggest privileges of my life to know Simon and all the people that I came to know through him and all the projects that we had together. And I want to thank the people who are here who are part of those. And thanks to Simon. And thanks to you for listening.
Thank you, Claire. So we've got one more talk now before coffee break, and our next speaker is Nigel Minton. And Nigel and Simon go back quite a long way, although in terms of projects, there's probably just the Max Bio one I think that you closely collaborated on. But Simon was always keen to meet up with Nigel. They were partners in crime at many events, and I know that Simon was incredibly fond of Nigel, so over to you.
So, yes, this is a very difficult talk to prepare, really. So, I find this so weird. Anyway, what I was going to say is, before lockdown, in the year before lockdown, I must have done 6 international talks all over the world. Lockdown came, I've done 2 since. One was last week in Brazil. It's just not so easy as it used to be. And then, to give this talk, this is a very difficult talk. So, this is sort of my perspective. Is it the mouse that works or the buttons? Right. What I wanted to say was that I am a latecomer to academia as well. Nothing as spectacular as Simon. But I did my PhD in Aberystwyth and then went to the Centre for Applied Microbiology and Research, which is the old Biological Warfare Centre of the UK, and I went there in 1979. I spent 25 years working for the Department of Health, and then I kind of had enough because I went there to work on Clostridia. I was getting EU funding to work on solvents. We call them biofuels now. And they said, this is a Department of Health Institute. You cannot apply for this money anymore. So I was working on Clostridia to deliver therapeutics to tumours. And they said, no, we're an infectious disease institute. You can't do that anymore. So then I started working on C. difficile. Thought that would be important. But then the Department of Health said, no, that's not important. So I thought, sod that for a game of soldiers, and I went off to Nottingham, which is when I went to academia. I always like to show this slide. This is the incidence of C. difficile in the UK when I went to Nottingham. It wasn't important, apparently. But anyway, that's by the by. So, when I got there, lots and lots of opportunities opened up. And within 4 years, the UK ran this competition, which we've just heard about from Claire, for a bioenergy centre. £10 million for an actual physical centre and 4 programme grants of £2 million. So, as we've heard from Claire, Simon got one along with Simon Cragg to work on the Gribble. I got one to work on making biofuels using Clostridia, and Claire and Paul Dupree, who's here as well, also got one, as Claire has said. And Nottingham and Rothamsted tied for this centre award, and typical of the funding council, they couldn't make a decision. So there was lots of to and fro-in. They locked us in a room in Cambridge one day and told us all to come up with a solution. And the solution that they imposed on us, no one agreed with, and we went away. And then it was decided, no, we'd have a virtual centre, and all of these groups would all work together on what was called Busbeg. And this was when I first encountered Simon McQueen-Mason. So we've also heard from Claire about these frequent trips to Hanoi. Now, my recollection is that we had to go 3 times to Hanoi in order to get this small grant which Simon led, which was to have a student come over to our labs for 8 months. And this is the first meeting I have photos of, because also what you realise is that we didn't used to have mobile phones in those days, so we didn't take pictures all the time, so it was quite rare to have pictures. And this is a trip we made in February 2011. It might have been the first, it might have been, no, it probably was the second. I can't remember. All I know is it was bloody cold. And you could see all these people are wearing coats and warm hats because it was cold. And you can see it's so cold that Simon is wearing shoes. And that's extremely unusual to see Simon not wearing sandals. And it was also, they kept losing power in the restaurant, so there's power cuts going on here. So it was quite a strange experience. And then, shortly the same year, there was a big meeting in India to try and sort out some projects that the UK and India could work together on. Is this a pointer?
I think there is.
Is it red?
At the top.
Well, I'm colourblind, so there's no point in saying that to me.
Press it hard, though.
This one.
Oh, it is, right.
Yeah.
Okay. So we went there and we had speed dating out in the, this is in New Delhi at ICGEB, and we all gathered together and we all had flip charts and we tried to decide what we were going to do together. And then we had this group photo, and you can see that Simon is there in his sandals and his short-sleeve check shirt, his standard gear that Simon used to wear, all shorts obviously. And then after this project, after this meeting, we actually came up with a project which we called Rice Fuel, which I led, but Simon was on that project as well. And that went on for 3 years. But we were back again, as Claire's just shown, in Hanoi the next year in February. And this was to seal this deal where we were going to have these students come over to the UK for 8 months, I think it was. And as Claire said, Simon's actually wearing a shirt and tie here. And he's signing the agreement between MOST in Vietnam and the BBC in the UK. But obviously shortly after this meeting, Simon's back in his sandals and check shirt, shopping down in Hanoi. And then, not too long after that, because the advantage of all of us being in Busbeg, that we used to get asked by the BBC to go off to all of these places around the world. And this is us in Buenos Aires in Argentina, where we went for a workshop. And Simon's in the shirt and tie again. This is very unusual. So we're around at the ambassador's embassy, but they don't have an embassy in Argentina because obviously we fell out with the Argentinians, so this is just what they call their embassy. I forget what its official designation was. But you can see there's Simon. He's got a drink in each hand. I don't know if you can see that. And he always used to take such wonderful photographs because he always had this brilliant smile. Unlike me, who looks like a complete imbecile in this picture. But Simon always looked good in pictures, always looked good. And obviously here he is back in these sandals again, going round. I think that was an opera house in Argentina. So one thing that happened at this meeting was I had just bid to have a synthetic biology centre in the UK, and I was desperate to know what had happened. And Colin Miles was there. And I'd just come back from Vietnam, and I've come back from Vietnam, I've flown to Buenos Aires, and my ears were blocked up, especially on one side. And Simon convinced me that Colin had told me the outcome, but I couldn't hear. And he said, he told you. You walked down the street, you know. He convinced me. But obviously he didn't. He was just having me on. And I guess at this sort of point in time, our interest slightly diversified. I became interested in doing something about actually using that CO2 that Claire showed on her slides, actually getting the bugs to grow on that CO2 and make things, as opposed to using sugar from biomass. And at that time, I think that was about the time I got this centre, and that was the focus. And also I ran a network like Simon. Simon had one on biomass, and I had one on C1, on CO2. Use CO2 as the feedstock. And I guess not too long, during lockdown, I thought, well, it would be really nice to pull these two areas together. So came up with a bid to work with Canada, and the UK are very keen that we work with these countries outside the EU because of Brexit. And so we have this project with Canada, which is a 4-year partnership with Canada. It's called No Carbon Lost, and what it essentially says is, let's stop CO2 emissions when we ferment things. So when we make biofuel, we make CO2. Well, let's stop that happening. So it's called No Carbon Lost, and all of that carbon will go into your product, which is good. So we have this with Canada, and then I also have a transitional one. It's the same theme, no carbon loss or no lost carbon, that's cool. And just this whole idea was seeded by a workshop we had in Brazil, which Simon came on, and here we see the night before the workshop, and we're all happily drinking caipirinhas at some nice restaurant near Campinas, and Simon got a little carried away at this dinner, and he was blooded, and on the bus going home he was just absolutely asleep. He was comatose, and he got up at 5 o'clock and he wrote his talk, and he gave the best talk I'd ever seen him give. It was just a brilliant talk, and he must have still been slightly under the influence of alcohol. And then before that workshop, the Nottingham crew had gone to a Posada birding, and after that workshop, the York crowd went off to the island that's close to Sao Paulo, and these are a few WhatsApps that were sent to me. Obviously involve caipirinhas quite a lot, and snorkeling for Simon, and then I found this picture, which was, I thought it was the last picture of us having a drink, which was at the midterm review of these India projects, which was in just before lockdown, just a few weeks before lockdown. But it wasn't actually, because we managed to have a BBNet conference in 2021, in November, and the first person I met there was Simon, and he just came up for the day. And then we had dinner, so that was the last drink. So I guess, but what I would say is that academia is dominated by, by megalomaniacs, really, egotistical megalomaniacs. It really is. Whereas Simon was an incredibly gifted scientist, and he was just a good, honest bloke. He was a scholar and a gentleman. So.
So our next speaker is Joe Ross. Joe is the director of the Biorenewables Development Centre, Joe was really important working with Diana at the very start of CNAP, and was really kind of helpful for me as well, to me, when Debs and I moved up from Cambridge. And so him and Simon go back a long way. And so from CNAP, Simon was also involved in the Biorenewables Development Centre, which I'm sure we'll hear about from Joe.
So I first met Simon in the summer of 1999, and I've put up here a very dodgy picture actually of what used to be called the Plant Lab, which was built for the plant scientists really in the biology department, with some help from Unilever, as I remember, and that happened before I arrived. But I remember coming in the summer of 1999 and being interviewed for a job to work as Diana Bowles' lab manager. And the two people who interviewed me were Diana and Simon. And they interviewed me, and I was lucky enough to get the job. So about 2 months later, I came back to start my first day, and was very nervous. I was moving my entire family from Bristol up to York, and I was thinking, is this going to work? And I bumped into Simon in the corridor, and he said, "Oh, hello. You're new, aren't you?" And I thought, he doesn't remember me at all. and it dawned on him then that he'd interviewed me. And instead of saying, "Oh, I'm really sorry," or anything, he just leaned in and gave me a huge hug. And all the nerves went away, and after that, I knew it was going to work. I knew it was going to be all right. So we worked here for about 3 years, from 1999 to 2002. And I dug out from one of my millions and millions of emails, this is just slightly randomly, there's not meant to be any great significance here, but it was just to show that was the CNAP Science Board meeting on the 12th of March 2001. And you can look at the names there, and you can see a lot of those people are here in the room. So at that point we had Diana, Ian and Simon as the chairs in CNAP. Di Rees and Peter Lilfers as the advisors, and Dale, I think, had an advisory role there as well. Myself and Maggie, who were kind of helpers and have been helpers, I suppose, ever since in various different ways. And then Helen and Judith. And Judith, of course, has stayed working with Ian all these years since then. So lots of sort of continuity there. So I just wanted to show that one. I thought that was significant. So Simon and I used to be the two people that used to get in really early on a morning in the plant lab, on our floor anyway, and John the cleaner. It was the two of us and John the cleaner. And that was where I really learned about Simon. We used to get in, and then he would regale me with all these wonderful stories about Penn State, about cycling, about all sorts of different things, as well as some science mixed in with that as well. But I really felt I got to know him there and sort of fell in love with him, really. He was just such a lovely, lovely guy. So, moving on, 2002, we moved, and the big new development, which I knew was happening because Diana was writing the GIF bid when I first met her in 1999. But we all moved to these fantastic new laboratories in what was the new part of the biology department. And that was when CNAP really expanded then. We had a lot more space, we had more people coming in, and it was a fantastic time. It was, I just really loved and have always loved this idea of, seeing that fundamental research and then seeing it being applied, and that's always the side of it that I've gravitated towards. So when we moved across there, I was in this office here with Lot, who's also here today, and Simon, I think, if I remember it rightly, was kind of in this office here. So our early morning discussions continued. We were still both the first ones in on a morning. I think it might still have weirdly been John the cleaner. But then John eventually retired, and we had other people come in, and then the whole group grew around there. So Ian was on the floor below. I think Diana was on M2, that's right, isn't it? I think. And so we kind of built up that team of people, and things continued from there. And that was a fantastic time, and my memory is that it was really when we moved to that new building that Simon's interest and involvement in the whole biofuels area and bringing his plant cell wall expertise to biofuels really started to expand and start to flourish as we went through the noughties, or whatever it is it's called. But there were other strands to this as well, and I wanted to just share some of those with you. So Simon was, as we all know, a very, very keen cyclist, and he really introduced me to cycling, and road cycling anyway. And a lot of those discussions that we had in the plant lab and then in the new building were about cycling and his experiences, good and bad, with cycling. And we used to go out on a weekend, and it was Simon and myself, it was also Ian Graham, Jared Cartwright and Jerry Thomas. There was the five of us that used to sort of cycle a lot together in various different combinations. We used to do two runs. We used to do the Stillingfleet run, and we used to do the Bulmer Bank run. And this isn't a picture of us. This is just a random picture that I managed to find on the internet, which gives you some idea of how steep Bulmer Bank was. So here you are, right down at the bottom. and you used to come up, and it was an absolute killer, and you'd get up to this point here. Now, believe it or not, there was a time when I used to be able to kick Simon's arse on a bike. Okay? I really could, over this distance, and that absolutely changed. But there was a few years when I was fitter and there was slightly less of me than there is now, and basically I used to get when it was just Simon and I used to get up to the top, because you basically finish in the little village of Bulmer, and then I would get off and wait, and then Simon would come up, sort of wobbling up to the top, and I would just be there smiling, looking at him, and he'd be like, But then things started to change, and I remember one particular occasion when we made our way up and we more or less stayed together, and I'm thinking, he's staying with me, and we got to the top, and I was absolutely wrecked. I got off the bike, I got down on my hands and knees and basically threw up on the grass verge, and Simon just stood there. And he told me years later that was one of his finest moments. I just absolutely loved it, because he'd gone through so much pain. And this was payback, basically. He was able to get me back. So, Simon was very much responsible for introducing me to that spell of my life before I found golf, when I was a cyclist. Yeah. So the other thing Simon persuaded me to do when I turned 40 in 2004 was to go on what he called a veterans cycling race. Okay, and this was for over 40s. And he said, we'll be fine. Come on, we'll go into it. There's over 40s, but there's also over 50s and over 60s. So, we might do all right in this. We've been doing all this kind of thing. So this is us before the race. We the two of us lined up. Somebody took a photograph of us, because there was only the two of us that did it on this occasion. And we went to the start, and they were setting us off in batches. So we were in the over 40s group. And then I think about 10 minutes later it was the over 50s, and then 10 minutes later it was the over 60s. So we set off, and immediately we were left behind. Everybody else in that race who was over 40 was shit hot. They were really good in comparison to us, and we were the total duffers in comparison. So we were desperately trying to keep, as this little dot disappeared in the distance, and then there was a kind of thunder, and the over 50s came along. I just went straight past it. And then the over 60s went past it. And it was the worst of it wasn't a single straight line, it was a circuit. It was a 7-mile circuit, which he did 6 times. And so every now and again, one of these groups would come and just go past us, and they would be banging on trying to do it. But anyway, that was a great example of the kind of things that Simon introduced me to, which was fantastic fun, but we didn't meddle, as they say, in that particular one. So the other thing that we really did a lot of was this, okay? We did a lot of walking, and some of the photographs that Ian's shared are from these same occasions. So we used to go walking at the end of January in the Lake District, and it was almost always the 4 of us that went. So Mike Roberts, who was there in the plant lab when I first came to York, shared an office with Simon, myself, Simon, and Ian Graham. And I've kind of tried to think about those trips and think about what we were all particular for. I was the organiser. Okay, it was my job to kind of cajole everybody to go again and to book a place for us to stay, because we would go on a Saturday morning and come back on a Sunday evening. So that was my main function. Mike was the pathfinder. Once we got there, he was the guy that was in charge of all of the maps and basically, made sure that we went the right way, and he was really, really good at that. Ian was, I would describe as the counsellor. There was always an occasion on the weekend when he would be walking, having a very serious, very in-depth, usually scientific, but sometimes not, conversation with each of us about some aspect of our lives, usually work lives, and Ian's always been, in my opinion, fantastic at that. And Simon was the swearer. and Simon was a really, really good swearer. He had that ability, and I think it sort of came out when we were away in the middle of nowhere like this, and he had a lot of space to swear. And he could swear, with curiosity, he could swear with anger, he could swear with kind of fun, but it just poured out of him. And what used to happen on a Saturday night was that we would go and we would stay in a particular place and we would have a fair bit to drink and we usually play cards. And Simon always used to lose at cards. And the swearing would absolutely pour out of him then. And we were often in these places with these little couples that were trying to have a nice little meal. And then Simon was just tearing into it, tearing into us with his swearing. So we went for several years, and you can see these are different pictures. I don't think we started in 2004. I think that's when I first had a digital camera, so my pictures go back to then. But we went for several years, and we went to several different locations, almost always in the Lake District. And these are just pictures of us on the top somewhere, smiling away. And you can see Simon's famously bought the dog on more than one occasion, which was tricky because there's a lot of sheep in the Lake District, and he spent a lot of time tearing after the dog, which was tearing after the sheep. And that was the last walking trip that we did that I had a picture of in 2009. But one of the things Simon was famous for as well was that he was really fast. He's got these great long legs, and he used to go up and he used to go down much faster than the three of us. And we went up one year to Helvellyn, and basically went up, and then there was a huge plateau, and then you went up again. And Simon went off ahead, and the three of us got up there. And when we got there, there was this huge plateau that was like a mile wide, and he was nowhere to be seen. And we just thought, where on earth has he gone? And we hunted, and we finally found him in a sort of a bit of a depression that you couldn't see from where we were looking. But that's my memory of him as well in the walking, was that he used to go and always be ahead. Usually by the Sunday he was kind of bitching about the fact that he wanted to go home, which was fair enough. But Simon's best swearing was when we did this mountain. This is Tryfan, which is a famous mountain in north-west Wales, in Snowdonia, I guess. And we went there one year. We only went 1 year to Snowdonia. But we went up, and for some reason we ended up going up a much steeper side of the walk than we intended to, rather than going up the sort of easy way to go up Tryfan. And it was at that point that really we started to realise that Mike and Ian were quite confident, had a good head for heights, and Simon and I really, really didn't. And I just went very, very quiet as it started to get quite, like we were going to die, basically. But of course, with Simon, what came out but just more and more and more swearing. And the bit I remember the most was there was a point where we'd kind of got up to a point, and then there was another bit that we had to climb up, which was too high, and you weren't able to reach where you had to climb to. So Mike assessed this and went round what I regarded as basically a suicidal ledge, and appeared at the other side, but was quite confident to be able to do that. But then Simon was trying to get this help to be lifted up, and he didn't think that Mike was helping him fast enough. And at that point, there was just an explosion that kind of filled the whole valley, as I remember it, of Simon swearing. And I try to say this without swearing, but boy, oh boy, he really, really let me up on that occasion. Anyway, we all survived, we all got down, and that was a good thing. So that was my memory of those kind of occasions. And there was all sorts of other stories, but that was probably the most memorable one, certainly for me. So I was asked to talk about this. I haven't got a lot of slides altogether, but I'll just tell you a little bit about the Biorenewables Development Centre. For those who don't know, it's a subsidiary of the University of York. It's focused on scaling up the ideas that come out of the university research. And in 2008, we started to discuss this idea. I'd, by this time, become a business development manager in the university's tech transfer office. And Simon and James Clark from Green Chemistry both won £1 million, each won £1 million. And so Simon, based in CNAP, and James Clark, based in Green Chemistry, decided, and with input from myself and from Mark Granow in particular at that stage, that what we would do would be set up a combined scale-up facility using those two £1 million sums, that we could then make a start on having some kind of scale-up centre and see if we could take it further. And Simon played a really pivotal role in that. I mean, he was already really keen to do more applied things, but this was the opportunity for us to take things forward from there. So we had a number of other people join, Debs Rathbone joined, and gradually we built up the team that had a focus on both biology and chemistry, but taking it, as Ian best describes it, one step out of the lab. So you could take things, you could try and get them to a stage where industry would be interested and actually try to apply some of the things that we were doing. So this is just a fairly random collection of pictures really, but we set up the centre in the end after an initial attempt to do it out at Ferro, which didn't work for various reasons. We decided to do it in the science park, in the bio centre on the science park. And initially we just took these 3 offices, which we converted into labs and where we had our scale-up facilities. And looking back on it, it was a bit of a bonkers place to do it because it was the top floor of a multi-occupancy business incubator, which was just like normal labs. So we were trying to shoehorn all these pieces of equipment in. And those 3 labs are kind of this little bit here at the end of what is a big L-shaped building. But this was a fairly typical piece of equipment that was too big, and you can see we had to take out the ceiling tiles to get it in. But this was our ammonia fibre explosion machine, which Simon designed really. And we worked with an engineering company and we built it and we put it on the top floor of this building. And that was where we were then able to do a whole series of experiments, basically exploding open different types of lignocellulosic material so that that could be the first step in terms of processing them. Then with a lot of help from Ian, we got another batch of money. So this is what we call the processing equipment 2. And this allowed us, as well as this equipment, to buy a lot of equipment more on the biological side, so DNA sequencing equipment, liquid robot handling equipment, and some of that went here, some of it stayed actually in biology for a while, and then eventually we moved it across here. But there was an evolution as all of this started to develop. We had a BDC board, and Simon was on that board right from the very, very start and sort of advised us all the way through as the whole centre developed. And then eventually the sort of penny dropped and we realised that we shouldn't be in a multi-occupancy lab, we should be on an industrial estate. That was a far better place for us to be. So we moved the whole facility to this industrial estate, which is based on Dunnington, which for those of you who don't know is just slightly to the east of York, just outside the outer ring road. And that's where we are to this day. So all of our facilities, all of our sort of biological facilities and some analytical facilities are in here. We have a chunk of this building, we have a chunk of this building, we have other bits and pieces that aren't on this site, and we now have a team of 32 people, and some of them are here, and some of these people are people who are there now, some of these people who've sort of worked with us and gone. You can see Maggie there, Mark, Debs, Anna, Elspeth, Joe Bennett, myself. I took a much earlier picture of myself, just a total vanity trip there. But this is James Clark as well, who was the head of green chemistry that I mentioned, who was really pivotal in setting the whole thing up. And this happens to be the picture when you go onto our new website now, because we've also now started to try and integrate people working in the university and working over with us. And of course, the poster boy for that concept is Leo Gomez, who now appears on the front of our website with Rosie. So that was really all I wanted to say. I think Simon's had a huge impact at York in all sorts of different ways, and we've heard a lot about a lot of the research he's done internationally, but I think locally as well, in terms of inspiring a lot of people to, including myself most definitely, to take this applied route and try and actually develop it further and to do big things. I mean, a lot of my boldness, I think, actually originates with Diana Bowles. I think she was the one who really gave me that inspiration to be bold. But it's absolutely, as many people have said, is also something that Simon had running right through it. And one of the things that James said on hearing of Simon's death, which I thought was lovely, he's just said he was one of the good guys. And I think that's exactly right. He was a fantastic person to work with. And I think a lot of what we've got here and the facilities and the people and all the kind of culture that we've got is very significantly down to him. That's it.
Thanks, Joe. So our next speaker is Alexandra Leno, and Alexandra is a senior researcher in CNAP and worked with Simon for many years, and is going to tell us about a project that I think is really a good example of just how creative Simon was in terms of developing projects and ideas. Alessandra?
Good morning. Thank you for the introduction. So it's not about biofuel, and I never went to Hanoi. So I started working with Simon 15 years ago. I was coming back from traveling, and I got a very small project looking at the feasibility of converting waste paper into biofuel. And looking back, and that was a test of what to come. So I contributed to many of the projects which have been presented today, and then did a bit more economic feasibility looking at making biofuel, but from agricultural waste. And everything changed in 2004 when Miriam from the Royal College of Art came to visit us, and she wanted to make new biomaterial. She wanted to use agricultural waste, and she wanted to use circular principles. And so Simon got inspired and remembered that a long time ago he had a collaboration with Professor Mike Gidley, working on bacterial cellulose composite, and to understand the role of expansive. And so he said, we should try to make bacterial cellulose. So we got the strain, and yes, we demonstrated we could use wheat straw to make bacterial cellulose, which is on the top right. And so this is when the whole project changed, because on top of making biofuel or biochemical, we started to make biomaterials. So this led to a proof of concept where we tried to use the bacterial cellulose to make textile, but that was not too conclusive. And so we moved to a longer project where Daniel helped us isolate a strain, and we tried to make cellulose to actually produce textile. And quickly we realized that we had to do more and more, make more and more cellulose, and so we didn't have the right equipment. And that's when Simon started mentioning the pebble polisher. So when Simon was a young boy, he used to collect pebbles from the beach and take them home and polish them on his pebble polisher, and he wanted to get one. We ignored him. But he went on and on for months until he told us he bought the biggest pebble polisher that Britain could sell. And so we had to adapt it to be able to use it in the lab, and this is what we did. And we started us purchasing quite a range of equipment. So we do have a washing machine, a clothes dryer, we have a dozens of pizza dough box, we have hot chocolate dispenser, and a wide range of equipment. And he was very keen to discuss characteristics for all of this. And I think he spent hours with Heather choosing the washing machine. He really liked that. But then the collaboration with the Royal College of Art moved to an even bigger project, and this was, and we got funding for a textile circularity center, where and the last two projects allowed us to transform our biomaterial in textile fibers, which was very exciting and which was a move. But he also allowed us to move from agricultural waste, municipal waste, paper waste to textile waste. And because textile also make of cellulose, like the cotton and the viscose. And this works quite well, so we developed this circular cellulose concept, where we would take a range of waste which contain plant-derived materials, the straws, the clothes, or a genuine green bean, and we used enzymes and bacteria to make our biomaterial, bacterial cellulose. And as you can see now, it's quite pretty now. It's nice, it's white, it's pure, and it's devoid of lignin, devoid of any cellulose, or even of textile dyes. We demonstrated we could transform it into fiber, which we want to move into yarn and fabric to manufacture regenerative textile, which at the end of life we go through this cycle again, which is very nice. But the textile circular center also gave us big insight on textile. And the textile sector has a lot of bad press, and there's a reason for it. It has a carbon footprint similar to aviation and shipping. It employs a lot of people, mainly women, who are very badly paid. But what was more surprising from our point of view is that it's a very complex and very linear supply chain, which means that if you look around you, you are wearing clothes made of cotton, which have been blended with polyester. These have been dyed, then they've been weaved, then they've been transformed in garments with button and zips. You're going to wear them, and 80% of it will just end up in the bin. So this is a waste. So there is some recycling, so your clothes can be transformed into rags, or they can be put into insulation, or if you're lucky, they might go into a second-hand market system, and then being binned. But less than 1% of our clothes are recycled into new clothes. Now, the concept I had presented just before was circular, so we said, oh, we've got something which might interest people. So then we started trying to develop the idea, develop the process through proposal, and that's the one which got funded, but we wrote about twice that. And Simon loved writing proposal. It really was his favorite activity. And it was an honor to develop ideas with him. But for all his research interest and scientific mind, Simon thought admin was done by fairies. None of his projects would have gone through without Jane. Managing budget happened because of Claire, because of Julia, and Alison. We did start collaboration agreement, and somehow they got signed. And Simon had no interest in non-disclosure agreement. Not at all. So this was quite busy time, but we didn't do it alone, and we worked with lots of people. And Simon liked people, and people like Simon, because he could relate to everybody. That you be a student, a professor, or some big industrial, he could relate to you. He would tell a good story, a story about himself, or a work story, and I still remember him speaking about the active site of cellulase to people who had never done biology. And they got it. So to his staff, he showed a lot of empathy, and he was very patient. And he just let them be. To his collaborators, he used to have a vision, and so they just wanted to work with us. It was very good. And to the students, he always gives them a chance. He came from humble academic background, and anyone who wanted to come in, he would take. Everybody knows that he had not many degrees, but he did finish with a first degree, and he liked ambition, and he would support ambitious students. Visitors, he welcomed anyone, especially from the Isle of Wight. Anyone who wanted to come in our lab, that they've done biology, he should say, yeah, go on, just tell Alex. So we spoke to a lot of different people, which was very exciting. And yes, we were developing, no, we don't say, we were pioneering an innovative bio-based textile recycling technology, which would revolutionize the sector industry. And having impact and spinning out his research to something which would add value, which would make a difference. He was very excited by that. And he would have loved it now, because we have all this big fashion company coming to speak to us. And he would have loved it. So how was it working with Simon? It was challenging, because you had to keep the pace, but it was really inspiring. It was fun. It was definitely fun, and infuriating. Yeah. And on a spiritual note, this is a picture of the Isle of Wight. That's where Simon called home, and that's where he retreated when the pandemic started. So all what I've spoken about didn't involve any cocktail. It was mainly done through Zoom. But as soon as he had updated his Wi-Fi connection, it's a place where he could write, it's a place where he could collaborate, where he could do what he wanted. But it's where he struck the right balance between work and life. And importantly, that's why he could get very easy access to the countryside and the sea. And I was fortunate that I went to visit him there for a holiday where we camped on the farm. And he seemed very content. And you could sense that really he got it sorted now. And that's how I want to remember him. So.
Thanks, Alexandra. So our next speaker is Simon Cragg, and Simon and Simon came up with the concept of the gribble as a source of enzymes for lignocellulose processing, and Claire previously mentioned about how these became the pin-ups, I should say, of the biomass world, but particularly the BBSRC, who just seem to love them. So we'll hear a lot about the gribble, I'm sure, from Simon.
Thank you for the opportunity for me to recognise what Simon has done for the team that I work with. So I'm speaking on behalf of really quite a lot of people about the impact that he has had. So sometimes I'll be talking about other people and what they've achieved, and the underlying message is that that was all facilitated by the sorts of characteristics that you've seen come up time and time again through people's presentations. And here is a map. I resisted the temptation to animate it. It's to give an idea of, so starting at the top, the impromptu seminar, I'm guessing that was about 2007 or 2008, and down to the bottom, things are still moving forward and expanding. So the first step, the impromptu seminar, was prompted by a phone call from Simon's postdoc, helping to do some exploration on Simon's behalf. Simon thought there must be some other organisms out there which are able to break down wood, and had come up with the idea that there were marine creatures out there which do it. The postdoc, King, contacted my PhD student, and I tried to politely not grab the phone out of her hands, and had a really interesting conversation with Simon. I'd been making a career of looking at these animals as a problem, destroying the sorts of piers we saw on the Isle of Wight. That is a textbook case of the sort of damage that these creatures can do. Anyway, we're talking with Simon. I realised that these creatures had another possible role. They could be a part of a solution rather than just a problem. And so I happened to be taking my in-laws for a holiday in Yorkshire and said, well, perhaps I could drop by and expand on this chat. And found suddenly ushered into a room with august company. And I think we've got Ian and we've got Diana here, and had Simon there. And it was a fascinating opportunity to think off the top of our heads. And I really enjoyed exploring the ideas with them. And as a result of Simon's thinking that there's stuff out there to be discovered, as a result of the fact that Simon's comfort zone was much wider than most people's, he's not afraid to go off into areas. So here we have a plant physiologist who would take on spider silk as something worthy of study. And so these small marine borers were something that he could happily tackle. What I discovered was a man who was laid back, but focused on ideas, ready to grasp ideas. So he'd be looking sort of abstracted, and suddenly would come up with an idea to follow up. And someone who was open to suggestions, so he was not, didn't feel that his ideas were the ones. He would want to hear what other people had to say. And I quickly realised that he was a phenomenal team builder, and had great pleasure working with lots of members of the team. Delights through happy meals together, working in the laboratory one-to-one, and so on. And later on, I came to realise that Simon was an absolutely extraordinary networker. And he did it not for kudos, he did it to make things happen. And underneath all of what I want to say to you is the feeling of Simon as a mentor. And he was a mentor for some of the people I'll just highlight briefly. But he was also a mentor for me. He didn't set out to do it, but I found all sorts of my practice were affected by what I saw. So stepping through this diagram, following this extraordinary seminar, was a proof of concept funding from Amazon. I hope I got that spelled correctly. Funded work in between people in Portsmouth and Simon's team to prove that this marine borer concept was feasible. This led on to convincing BBSRC to release some funds for a serious project. And rather than the initial target for what we were doing, which was shipworms, which bore huge holes into wood and nearly prevented Columbus completing his voyages, we were working on a little creature which I'll probably call a gribble, and as a scientist I should call limnoria, hence the name Limnoromix. We were learning from Simon about how you could mine the genetic information, look to the proteins and so on. Branching off, we've got the experience we had through Busbeck, and I can remember many interesting meetings, and I also got the impression that people smiled when a picture of a gribble appeared on the screen. They were charmed, and then got back to the day job, but our animal helped us. As the project developed, there was an opportunity to go to BBSRC for funding to help us build links with the USA, and that was very much on the back of what was being achieved through the Limnoromix project and through Busbeck. That led on to links for us with the National Renewable Energy Lab in the States, and eventually in Portsmouth the establishment of a Centre for Enzyme Innovation with a focus on enzymes which break down tough materials. The big focus there is on plastic, but we continue the interest in woody materials. At the end of that partnering, we had a meeting hosted by the Linnean Society, which was a great way of getting our American collaborators over, they loved the august surroundings for this. And it helped shape our interest in the tree of life, the diversity of organisms which do this breakdown. And on the other side, you will see a trajectory of LBNet and BBNet. And again, this was a learning experience. Simon was always looking at how the discoveries lead you on to things with practical applications. So, and the LBNet certainly gave us an opportunity to follow up a curiosity. Working with our little borers, one of the things we did was count their fecal pellets, which is a specialist topic, and a conversation stopper at meals. And we got on to consider things at a heroic scale, which is the problem of racehorses. And the problem is they produce not tiny little fecal pellets, but great mounds of dung. And we'd got someone who had somehow thought that all we needed was an enzyme which could be produced somehow in huge amounts and could be poured onto piles of horse dung and converted into biofuels. Yeah, right, it's easy peasy. Well, we got LBNet to finance something to prove that it wasn't that easy. And the big step for us was the strategic Lola project, where we opened out our focus on various organisms of interest. So enough of the complicated diagram. So limneromics, little creatures off to the right, they're about so big, 3 millimeters long. They have a huge digestive gland, which is, yeah, I think I'll do it with the cursor here. I'm more used to doing that. So there is the digestive gland, and you can see that a lot of the inside area in the animal is a digestive gland. And that's because these creatures only eat wood, shown as the speckled material in the hindgut. Confusingly, the animals have a stomach located in their head, which would certainly limit the size of the meals you could have, but they just keep eating. And this diagram here is, and I think Yi Li's here, and that's, thank you for this remarkable insight. We're looking at a particular type of enzyme, a GH7, that bites off units of 2 glucoses of cellulose chains and keeps on doing it in sequence. Here we've got lots of fungi, lots of bacteria, a few protists, and an unusual group of animals. Down here, a few rare crustaceans. So, a puzzling thing. These creatures have got an unexpected enzymatic capability. And I should point out, led by Andrew King, and a sort of blending of teams. And this was really capturing the idea that Simon had when he sent out the inquiry in the first place. This led on to looking at the enzyme, and I've brought in here John McGeehan, who, with Marcelo Kern, led this particular paper. They're looking in detail at this GH7 protein, and with the colleagues in NREL, looking at how the protein operates, not just in crystalline form, which is the way that the structure is solved, but also in liquid. So, because we're coming up to lunchtime, I didn't want to induce seasickness, which is what I get when I look at this sort of image when it's animated, because the whole thing pulsates. And you find out which bits of the protein are flexible, and you see all sorts of interesting things. And if we look at this little graph, we're looking at all the GH7 structures which were known at the time of this publication, and in red are our creatures. And what this is telling us is that the surface of this protein is very acid. And we thought that this was something to do with this protein being able to function in salty conditions. And I thought, well, of course, it's an animal from a salty environment. But Simon saw that actually the interesting thing was in industrial treatment processes, the solutions involved get very salty. So this was spotting the opportunity there for biotech. Now, the Linnaean Society meeting, this gives you, this picture here, gives you one or two recognisable people. So Charles Darwin, I think that's Wallace, and we were actually in the room where Darwin and Wallace put forward jointly the idea of natural selection. And a nicely attentive audience, and who's in the front row? So this was clearly not immediately after lunch or immediately before lunch. Simon's absolutely on the case. And presenting in his usual laid-back but engaging style. So we had a nice breakout, and the Linnaean Society does a rather stylish library, and on the left you can see us looking suitably serious as we ponder on our questions. Simon off to the left there. And then a notable picture at a certain time of the evening where Simon isn't there, and there's a mysterious time where he disappears. You don't see it happen. And this sort of thing led on, and what I'm saying here is that this is a team of people who didn't work together in Portsmouth, who, as a result of this collaborative work, suddenly came together as a team. And the impact for us was down to the imagination that I've been talking about. Here's one of those people, a very lucky master student. He must have thought that science was easy. He tackled this delightful little creature, Daphnia, here. He got the protein expressed, crystallized, and the protein structure solved, all within a master's program. I thought, yeah, science is easy. And it launched him on a career. And if we look here, we're looking at the same protein I was discussing, two views. Red shows the acid surface in nice simple terms, and up top, blue is base and white is neutral. The Daphnia, living in freshwater, had a not very acid at all surface. Limnoria did have a very acid surface. So well done to Ali. and he'd moved onwards and upwards. Here we have Amaya, who came and worked here, produced the marvellous image here of the tree of life and how the ability to break down wood was spread across it. And she interpreted how in some of our creatures the enzymes were shared between microbes and the animal, and in our little creature it produced all its own enzymes. And sorry, racing forward, which is good. Ruben here also benefited from working with Simon's team and working on the shipworm, which was nearly the nemesis for Columbus. And we've had lovely publications coming out of that. And I'm sorry, Giovanna, we're going to pass over that because I think I've got one point to make before I can conclude. Pass on. So this is the environment within our animal, and we were able to find that the animal was producing its enzymes into a gut with very low oxygen levels, and the animal was, interestingly, just digesting the cellulose. Yeah, now this is something I wouldn't miss, or to read this rather than me read it to you. I was very touched by this. Graham was one of the people who came and worked here, and I asked him if he could give me some vignettes, and he wrote me a mini essay, and I extracted that from the essay, and the tears are prickling my eyes again. I thought I couldn't express it better. So finally, we're looking to the future and thinking about the fact that these breakdown processes occur in the natural environment too. In salt marshes, and Neil's had a student working on exactly that question, in mangrove ecosystems and in seagrass meadows. In all these places, higher plants are producing detritus, which is a vital part of the functioning. So there's biotech, there's also ecosystem level implications. So with all of that, I'd like to say, from all of us at Portsmouth, thank you to Simon for giving us this impetus which has led on to so many interesting things.
Okay, so I'm the last speaker. Unfortunately, a lot of what I'm going to say has been covered by the other speakers, so I will be able to be hopefully fairly brief. Okay, so Simon was probably the first person, or one of the first people in York to welcome me when I moved up from York, moved up from Cambridge to York about 21 years ago now it is. And we moved into those beautiful new labs that you saw earlier that Joe showed in his presentation. And we shared one of these big labs, and his office was kind of next door to mine. And as Joe said, he used to get in early in the morning, and I used to come in and make a coffee, walk past his office, and always stop. And we would have a chat, and we'd talk about everything and anything, and often put the world to rights. But some real nuggets of ideas came out and future collaborations. And one of those was around unlocking the potential of biomass to produce sugars you can ferment to produce biofuels and chemicals and to address climate change. And the slide here is one that Simon always showed when he was giving lectures to undergraduate students about just how wonderful plants were, and in particularly the plant cell wall, and how it could allow trees like the sequoia to grow to hundreds of feet high. But I also showed this here because I have to laugh because we decided we'd have a tree planting ceremony today to remember Simon. And so James gave Claire a call and wanted to know what Simon's favourite tree was so that we could plant it on the campus. And of course Claire said to him, it's a sequoia. And of course, it'd just make a statement, wouldn't it, if we had a sequoia tree growing by the department. So we've heard a lot about this. It's about, the plant cell walls are really tough to degrade, and one of the big bottlenecks in terms of production of biofuels from plant biomass for second generation biofuels is you've got to unlock the sugars here. And that's kind of what we were working on. We wanted to develop ways in which we could economically break open these cell walls to release the sugars that could then be fermented to produce biofuels and chemicals. And the concept is not new. It was, Henry Ford in the 1920s had cars that drove on ethanol, and there's a really nice quote there from Henry Ford that's saying that you could basically ferment any vegetable matter to produce ethanol, even sawdust. And we've heard a lot in talks today about agricultural residues and using them as feedstocks to make biofuels. And of course, wheat straw and rice straw are some of the obvious candidates. And we heard from Leo, and we heard from Claire as well about something that really incensed Simon. He was absolutely passionate about this. So rice straw, millions, hundreds of millions of tons of rice plants are grown, and hundreds of millions of tons of straw are burnt in the field. And it's generating black carbon that's bad for people's health. It's contributed to climate change. And he would say, but what a waste of a resource. This is a fantastic opportunity to produce, fuels and chemicals. And he got really incensed by this. So he actually raised money for a workshop, a number of workshops in Vietnam. But this one was specifically to look at the valorization of rice straw. And we arrived, and he was convinced that the Vietnamese government had told the farmers not to burn any rice straw when we were there. So we landed in Hanoi, and there's no pollution, air pollution or whatever. And what sort of reinforced this thought was that when we had the meetings, the scientists say, no, we don't burn rice straw, we do useful things with it. And so this carried on through the workshop. Anyway, we eventually, after a few days in Hanoi, left, and our flight was late in the evening. And Simon was convinced that the government had told the farmers that they could now burn the straw, and they thought we'd gone, because as we were driving to the airport, the fields were ablaze and there was smoke everywhere. And so he was absolutely convinced by this sort of conspiracy theory. So as you've heard, he and I kind of were really keen to explore the way in which all sorts of living organisms degrade lignocellulose. And we've explored cellulose digestion in insects. We've heard about the gribble, the marine wood borers. We looked at in microbial communities as well, in all sorts of environments like salt marshes. And we composted everything. So composted wheat straw, rice straw, sugarcane bagasse, empty fruit bunches from oil palm, and built up this sort of library of enzymes that we're exploring and characterising, and still will probably keep me going till I retire. And we've heard about BizBec, and this was a big opportunity where the BBSRC put in that it was the largest single investment by the BBSRC into any initiative to develop this sustainable bioenergy centre. And we've just heard from Simon about the marine wood borers and how cute they are, but also, depending on how you look at them, they can be pretty gruesome as well. But they are voracious wood borers. And what fascinated Simon McQueen-Mason, which Simon Cragg didn't mention, was the fact that these animals didn't have any microbes in their guts. So, termites, sheep, goats, cows will all live on a diet of lignocellulose. But they're dependent on the microbes in their guts. And so Simon, in his creative thinking, thought, well, actually, the gut of this crustacean, you could think of it like a sort of mini industrial reactor. And if we could understand what's going on there, the chemistry, the biochemistry, the enzymology that's going on, maybe we can replicate that and do it at a much larger scale. Oops. And some great science came out of it, and Simon just mentioned about this interesting enzyme. It's a cellulase, but what was fascinating about it was the first one to be found outside of a microbe in an animal. And yeah, another pin-up picture of the gribble there. I made the front cover of PNAS. So we worked a lot with microbes as well. This is my favourite fungus, Paracetosporium putridinus, that we identified in wheat straw compost. And it's fascinating because we're realising now through the technologies that we've developed that we know still a lot to know about how lignocellulose, how wood, plant material is broken down in the environment. And we're still finding new enzymes with new activities that have commercial value. And through this microbial work and through Simon's collaboration in Brazil, we had this opportunity to apply for UK-Brazil joint funding. So this has been announced, and we put in this grant proposal, and it did incredibly well. It was evidently the highest rated grant proposal in that round for the BBSRC from all their committees. And it was the very first one of these awards to be given. And so we got contacted by the Chief Scientific Advisor for the government, Sir John Beddington, asking us if we would go and accompany him on a mission to Brazil to meet the Minister of Agriculture and so on. Simon couldn't make it, so I went on my own. And so that was a really successful visit. And we had this lovely collaboration over a number of years with Igor Polikarpov, who was part of Simon's big Brazil project. And so it meant I could tack on our project meetings to those meetings that Leo showed in Marasias, and that beautiful beach resort. And I just wanted to show this slide because it's a bit of a contrast. It wasn't all sun, sea and sand. The weather was bad sometimes, and there we are discussing our project. So we did do some work there as well. And this work was really important, or the work that's going on in this area is important because we need to produce biofuels from plant material if we're going to escape or realise net zero and move away from using fossil fuels for liquid transportation fuel. And Brazil is the second largest producer of bioethanol after the US, and investing lots of money now into these second generation ethanol facilities. And what you can see there is, so what happens, so all the bioethanol in Brazil was produced from the sugar from sugarcane, and once the sugar has been extracted from the cane, you're left with bagasse, and you get mountains of this stuff, which you can see there how high it is compared to the size of the trucks. And so this is, potentially an incredible resource if you can unlock those plant cell walls, release the sugars to produce bioethanol. And so there's a lot of investment now in these second generation bioethanol plants in Brazil, but still that main bottleneck is the enzymes that can open up these plant cell walls. So there's still some really big opportunities there. Nigel mentioned the links with India. So because of BizBec, Simon was invited to attend that workshop that Nigel showed, and met there Shams Yazdani at ICGEB in Delhi. And Shams at the time was working on these rice stem borers. You can see the larvae there that cause a lot of destruction in the paddy fields. And there's some really nice similarities to what Shams was doing to the work that we were doing in York. He was interested in the microbes in the guts of the larvae and how they were digesting the lignocellulose. And so we decided we would collaborate. And as Nigel mentioned, we applied for funding. And the application was led by Nigel, and the project was called Rice Fuel. And it was a lovely collaboration between Nottingham, York, Oxford Brookes University, Shams in New Delhi at ICGEB, and colleagues at ICT Mumbai, and a number of other institutions as well. So this is all around, using rice straw and unlocking rice straw to the sugars in rice straw to make biofuels. That led to another Newton funded project, and Leo mentioned this, and this was pictures of us at a sugar mill in Hyderabad, and again it's about taking that sugarcane waste and converting it into not bioethanol this time, but into citric acid. That led to another big opportunity where we were looking at the way microbes could convert the empty fruit bunches from oil palm into biofuels and chemicals. And this is a real waste problem in Indonesia and Malaysia because of the vast quantities of palm oil that's produced. And these empty fruit bunches are really difficult to dispose of. And so, again, there's this, like rice straws, this incredible resource there that could be utilised. So I just want to finish. We talked about all the great science that Simon's done and initiated. He was absolutely devoted to his group. And as we heard from some of the other speakers, he was incredibly supportive to the sort of younger generation of scientists. And I just wanted to give Federico's story really as an example. So Federico was working on the marine wood borer project, the strategic S-Lola that we had. And he asked Simon if he could possibly start a little side project. And Simon said, yeah, of course, this sounds really exciting. And that project was to look at the way this insect, which you can see here, which is Thermobia, or better known as firebrack, very similar to silverfish, and how that digested cellulose, because it had been shown that it could also degrade cellulose in the absence of microbes as well. And using the sort of techniques that we developed over the years, these sort of omics techniques, Federico identified a new class of enzymes that hadn't been seen before in animals that really enhanced the degradation of cellulose. So he purified, clone expressed and purified the enzyme and characterized it. But what was really important and innovative as well is that Federico had identified that this family of enzymes were present in insects and also in insects that didn't degrade cellulose. And so he cloned and expressed these and found that they degraded chitin. And chitin is the material that makes up the exoskeleton of insects. And when that was knocked out, he found that the larvae couldn't undergo metamorphosis. They couldn't go from the larvae to the adult insect. So this really discovery, from looking at biomass degradation, unlocked a real unknown sort of puzzle in terms of how that sort of development occurred. And then he looked further into this family of enzymes and found that they're sort of spread right across the tree of life, not just in microbes where they're originally found and in insects, but also in important plant pathogens. And one of them's an oomycete and called Phytophthora infestans, and you'll know it as potato blight that caused the Irish famine, where 1 million people in Ireland died through starvation because of this attack on potatoes. And so Federico went on and discovered that this enzyme was absolutely essential for penetrating the plant cell wall, and showed that when that was knocked out with collaborators at the James Hutton Institute, that the pathogen could no longer infect the plant. So it's a really important target for controlling this pest. And he got 2 really fantastic papers out there in high impact journals. Simon would have been incredibly proud to have known that the department gave Federico an independent position, a lectureship earlier this year. And so it just goes to show just how supportive and generous Simon was with his time. And I don't know how Nigel got a copy of this picture, because I thought I had the only one. But this was I think our last trip with the 3 amigos travelling, and it was after Nigel's workshop in Brazil, and we went to this island off the coast of Rio and had a wonderful time. But we were really fortunate in that Ian and Sophia could join us as well there, and they were absolutely delightful company. And they put up with these 3 childish old men, and a lot of happy memories. And so with that, we're going to finish. And thank you for coming along. and that we've got lunch now, but there's a tree planting ceremony at, I think it's at 2:30. Erin's nodding yes, it must be, over at the biology department. So after lunch, if you want to join us, you're all very welcome. And then after that, we've got some refreshments in the biology department. So thank you all very much.